Hi everyone, thank you for joining us for today's webinar titled Improving Single-Cell RNA-Seq Workflow Efficiency to Scale Clinical Research. I'm Jackie and I'm your moderator for today. We like our webinars to be interactive, so we encourage you to submit questions for our panelists at any point during the presentations using the Q&A box next to your slide window. We will address as many of these questions as we can during the Q&A session following the presentation. Please note that all attendees are on mute. This webinar is being recorded and we'll send you an email when the on-demand webinar is available. A list of helpful resources and documents related to today's webinar is available in the resources list. Finally, to optimize your webinar experience, we recommend closing any unused browser tabs to maximize your bandwidth. And now let's meet our speakers for today. Dina Finan is a product manager at 10x Genomics. She received a Ph.D. in biochemistry at Stanford University, studying molecular motor proteins and their role in embryonic development. Prior to joining 10x, she worked in the field of protein engineering and antibody stability, and gradually made her way to product management. Also presenting today is Dr. Bhagyashree Birla. She completed her Ph.D. from Iowa State University in 2017 with a thesis focused on next-generation sequencing technologies. She has since worked in different life science organizations as an application scientist supporting various genomics products and services. She joined Azenta Life Science as a technical application scientist in 2020. In this role, she provides technical consultations for our customers to design their next generation sequencing projects and makes scientific recommendations to set them up for success. And first, I'll hand it off to Dina to get started with her talk.
Thank you, Jackie, for that introduction. I'm excited to kick things off today and talk to you about what single-cell sequencing is and how it can help you accelerate your pipeline with a much deeper understanding of disease and biology. I'll present a few case studies that highlight how single-cell resolution is really transforming clinical research, and then I'll end with some considerations of how to get started, particularly if you're interested in working with a CRO or service provider to help you streamline your work and do it at the scale that you need. So I'm sure I don't need to tell you that biology is fascinating and also very challenging, where incredibly complex systems and interactions arise from the simple fundamental building blocks that can be very difficult to untangle. And scientists need tools that provide this high level of resolution and characterization to be able to unravel this complexity. Over the last few years, profiling transcriptomes of individual cells has become an essential tool for characterizing heterogeneous samples. So, compared to traditional bulk methods, which are shown on the bottom row here, they analyze the RNA of an entire population of cells and yield an average of that measurement, shown in that stacked single bar, instead of representing each individual cell's transcriptome. So on the top row, if you analyze the transcriptome of a single cell at a time, which you can do with 10x Genomics technology, you can capture the heterogeneity of that sample. And this really enables you to catalog and profile your sample. We like to use this fruit analogy on the right to think about the differences between these two methods, and I think Bhagya will touch on this as well in her portion. But with legacy techniques like bulk RNA sequencing, it's sort of like sampling a smoothie that's made up of all the different fruits in a sample. So it can be delicious, but not too informative, especially if you've made a smoothie and you've put a banana in it. The banana really tends to dominate the flavor. So would you be able to tell all of the other fruits that were blended into that smoothie? No, probably not, just by taste alone. So the single-cell methods really allow you to maintain those individual components and assay them separately. As I already alluded to, some of these more traditional tools, such as qPCR, flow cytometry, and then bulk RNA sequencing, which I just spoke about, have really allowed researchers to come a long way in our understanding of biology. But as we move into the next era, we do need better tools with higher resolution. And there's actually a lot of similarities between these legacy methods and single-cell methods. For example, With qPCR, both methods involve creating cDNA from RNA that you isolate from a sample. And in both cases, you're analyzing differential gene expression. But they're different in that single-cell analysis not only maintains that single-cell resolution of the data, but also it's able to profile the whole transcriptome at once, as opposed to just a set of selected or targeted genes. With bulk RNA sequencing, in this case, both methods involve preparing an NGS library and sequencing of that library. And in this case, they're also both able to profile a whole transcriptome. But the key difference here is really that single-cell resolution is maintained. And then finally, flow cytometry is a very valuable and commonly used technique that has been around for decades. And similar to single-cell sequencing, one of the first steps here is to create a single-cell suspension from your sample in order to pass it through the flow sorter. Also, flow cytometry does provide data with single-cell resolution, but in this case, you're limited to the number of fluorescent antibodies that can be analyzed at one time. So the readout is just less comprehensive compared to whole transcriptome sequencing. And of course, I have to point out that these techniques actually work very well together in complement with single-cell methods. So they're often used hand in hand. And work using the 10x single-cell platforms has been showcased in published work that spans the fields of immunology, oncology, neuroscience, and more. And these publications have really, been featured in the highest impact journals coming from leading labs and research institutions all across the globe, and have really driven the field of science forward in an unprecedented way to cover over 3,000 publications today, and that number is just growing really rapidly. And besides a wealth of academic research, there are applications for single-cell analysis throughout the entire drug development pipeline. So in research and discovery, in terms of identifying novel targets, discovering antibodies that bind to specific antigens, to doing genome-wide CRISPR screens at single-cell resolution, and even to characterizing a complex cell therapy product. And moving into translational and preclinical research, there are really intriguing examples already in biomarker discovery, understanding the mechanism of action of a drug, and developing new model systems. And ultimately, of course, the end goal is to move closer to the clinic, to develop biomarkers to help guide therapies, and to be able to better stratify patient cohorts based on specific gene signatures or indicators of resistance. And here I'm just showing a few examples of some publications where our single-cell tools have been used to advance clinical and translational research. This is specifically in the areas of oncology and immunology. And I'm going to pick a couple of examples and move in order from left to right, just to highlight some really nice recent case studies. Just before I move on to those case studies, I wanted to just take one quick minute to look at actual single-cell data. And if this is a review for you, just bear with me, it'll be really quick. So this cartoon graph on the left is an illustration of a t-SNE plot, which is one way that scientists use to look at and interpret single-cell data. This is really a method of reducing complex data into a visual representation that's just easier to understand. And here each dot represents one single cell, and cells that are closer together in the plot have more similar transcriptional profiles than cells that are further apart. The cells can be grouped together into clusters based on those transcriptional profiles, and those clusters will have the same color. So on the right then is an example of a real t-SNE plot from a recent paper in Cell that you can obviously see has a lot more cells than the cartoon version. So in this experiment, the authors profiled over 1 million cells using the 10x technology to discern distinct cell types in the respiratory system and peripheral blood of COVID-19 patients, as well as healthy controls, to better understand the impact of the virus on these different cell states. So for the first case study, I'd like to start on that very left-hand side of the drug discovery pipeline. And an important application for single-cell RNA profiling is the discovery of novel new cell types. And this publication in Nature from a few years ago, that comes from researchers at the Broad Institute and Mass General, is a really nice example of how the discovery of a new cell type can be immediately useful to human disease research. So in the study, the researchers were cataloging all of the different cell types in the mouse trachea, which revealed this ultra-rare population that comprised less than 1% of all the cells. So as you can imagine, you absolutely need an assay with high throughput and high sensitivity to be able to detect this tiny population of cells. If you just sorted a couple hundred cells and profiled them, you would miss this tiny population. And what's really fascinating is that these rare cells, which they called ionocytes, actually turn out to express the vast majority of the CFTR protein that's found in the lungs. And this is immediately of interest to cystic fibrosis researchers who had previously largely focused on the ciliated cells of the lung for therapeutic approaches. When they performed a knockout in their mouse model that resulted in loss of the CFTR expression in ionocytes specifically, they saw disruptions in airway fluid and mucus physiology, which are phenotypes that are characteristic of cystic fibrosis. And so this is really exciting because by associating these cell type specific expression programs with key disease genes, they were able to establish a brand new cellular narrative for this disease. And now moving slightly further down the drug discovery pipeline, this paper, which comes from a really large group of researchers, mainly from the Broad Institute and Columbia University, is a really nice example of using the power of 10x single-cell CRISPR screens combined with looking at the whole transcriptomic analysis and cell surface protein expression at the same time. So this is truly what's called a multimodal or multi-omics study. And you might know that many relevant cell phenotypes are best understood at the protein level, and sometimes there can be differences between the RNA and protein levels of the same gene. This is why it's important to look at both. And you might already know that a key challenge in developing cancer therapies is resistance to immune checkpoint inhibitors. And while these inhibitors can produce really durable responses in some patients, most patients either don't benefit at all or develop resistance over time. And there are some known mechanisms for why this happens, but they really account for a minority of cases. So in previous work, before this publication, they had used 10x single-cell RNA sequencing to identify a specific gene signature that's predictive of intrinsic resistance in patients with metastatic melanoma. So in this study, they set up a novel screening platform. And what they did is co-cultured malignant cells that were derived directly from patient tumors, along with ex vivo expanded tumor-infiltrating lymphocytes from the same patient. So with these co-cultures, then, they could see dose-dependent and time-dependent cancer cell killing by the activated T cells. So once they had that co-culture system set up, they targeted 248 specific genes by CRISPR from the intrinsic resistance signature that they had previously identified. And then after that, they profiled the whole transcriptome as well as 20 cell surface proteins. And they did this on over 200,000 single cells. So at the end of the co-culture, by collecting the cells that had survived, they could see which specific CRISPR perturbations conferred immune evasion on the cells. So really, really nice model system here. And overall, their results suggested that the genes that were involved in T-cell mediated killing were concordantly regulated at the protein and RNA levels. But there were some exceptions that were seen, only at the protein level, which really, again, highlights the importance of making these measurements simultaneously. And then they went on to develop some computational frameworks to be able to model the effects of these CRISPR perturbations on the profiles of individual genes. And they were able to correctly reconstruct the impact of some known genes that were expected to be involved in immunotherapy resistance. But then they also identified some new and really intriguing candidates and pathways. And some of those, like CD58, for example, do not have a mouse homologue, which really highlights the value of doing these screens with human samples. And then for an example of using 10x tools in preclinical work, I wanted to highlight this paper on vaccine research, which was published by Sanofi in France. And I'll also just mention here that single-cell work has been used in some really excellent work on the COVID-19 mRNA vaccines to really characterize the immune response as well. But today I chose to talk about this work instead. So when you're developing vaccines, it's obviously important to have a preclinical model that can simulate the targeted age population and a relevant schedule for vaccination and boosters. And so for this study, they used the pertussis vaccines, which are very widely used. Most of us have probably had these. And they were looking at a unique mini pig model, a Göttingen mini pig. And they wanted to demonstrate the utility of this model system for these, types of longitudinal studies. And you can imagine there's many advantages of using these mini pigs over either small model systems like mice or large non-human primates. So in this work, they vaccinated neonatal and infant mini pigs with the whole cell or the acellular pertussis vaccine. And then they followed it up with the booster Tdap acellular vaccine. And they compared the immune responses of these different schedules of vaccination. And they used 10x Genomics single-cell RNA sequencing to look at the long-term maintenance of the immune memory cells and their functionality. And they did this 6 months after the booster. And what they found was that this model system did faithfully reproduce the cellular immune responses that humans do with the same priming and boosting program. So for example, they saw a stronger immune response when the first dose was given to infant pigs rather than neonates. So a really nice example of how you can use these single-cell methods in a preclinical sort of new model system development. And for this final example, I wanted to talk about a paper that describes the use of single-cell analysis in a phase 1 clinical trial. For a little bit of background, natural killer T cells have potent anti-tumor properties in mouse models, and they have been linked to favorable outcomes in humans with cancer. But there are very low numbers of these cells in our bodies. So in this paper, the authors from Texas Children's Cancer Center at the Baylor College of Medicine and ImmuneAI reported some interim results from the very first 3 patients enrolled in a phase 1 trial of NKT cells that were engineered to co-express a GD2 specific CAR with IL-15, which promotes the growth of these cells, in children with either relapsed or resistant neuroblastomas. And their initial results suggest that this strategy can be effective and safely expanded to clinical scale. And just to set up, what was really novel about this work, so in most polyclonal T cell products that are currently used for cell therapies, these NKT cells are largely absent. So you can see the low frequency on the column in the left on the set of plots there. They make up about 0.1% of PBMCs. And so that means that you really need new methods to be able to accomplish clinical scale expansion of these cells. And so what you can see in the middle column is that they were able to get the final products to a mean 95% purity for all 3 patients that they looked at. And previous to this, the most successful clinical scale expansion study only reached 54% mean NKT purity. On the right column is showing the percent of CAR-positive cells among the total NKT cell population. So here they're just establishing that they were able to scale these cells up and that they were well tolerated by the patients. So the graph on the bottom shows by qPCR that the absolute number of CAR NKT cells was quite different in each patient. So patient 1, which is shown in a black line, had the lowest number, and you can see that it was declining, whereas patient 3 in blue had the highest number that kept, the cells kept going up over the 4-week period that they were measuring. So by doing single-cell RNA sequencing on these pre-infusion products from all of the 3 patients, they were able to see 9 distinct clusters. So those are represented in the 9 different colors on the plot on the right. And this plot is showing about 9,000 cells, and they're overlaid from all 3 patients, which is just one way of looking at the data. And when they enriched for the GD2 CAR transgene, they could see that the expression was concentrated in one particular cluster, cluster 3, that had the highest level of that transgene. And that same cluster is also enriched in gene sets that are associated with central memory and activation versus exhaustion markers. And it's hard to see in this figure because all 3 patients are overlaid, but the frequency of cluster 3 was highest in the cell product of patient 3, which, if you recall, had the consistent increase over the 4 weeks. And so this could indicate that the persistence of these cells can be influenced by some specific characteristics, such as resistance to exhaustion. And so you can also imagine that being able to track these characteristics by single-cell RNA sequencing can help to characterize products before infusion in future trials. And so they went on after this to look at the therapeutic responses, and they found that these CAR NKT cells could infiltrate the tumors and mediate tumor regression. So there was some promising, very early results that in at least one of the patients, there was a dramatic reduction in the size of a metastatic lesion. So really exciting, work to show how you can use this high-resolution technique to really understand this complex product and, just help you be able to know what to look for. So now that I've convinced you of the advantages of single-cell sequencing and shown you some recent examples of how it's helping to advance drug discovery, hopefully you are as excited as we are to think about what comes next. So seeing a sample at single-cell resolution can really give us the ability to better characterize the heterogeneity of all types of tissue, no matter which area of research you focus on. And I don't have time in any detail to talk about our products today, other than to say that we deliver complete end-to-end solutions that just work. And they comprise a whole integrated workflow, including the instruments, the consumables, and the software for doing the data analysis and visualization. And another couple of pieces of the puzzle that I'm not showing on this slide are all of the sample preparation considerations that go into starting an experiment, and also our partnerships with certified service providers who can be really instrumental in helping you get started. And so before I hand things off to Azenta, I just wanted to touch on some of those considerations for preparing samples, especially for these larger clinical studies. So logistically, you might think about where and who is going to be preparing samples. Do you plan to collect them at many sites and send them to a central lab? And just thinking about what types of samples you need to be collecting. And then experimentally thinking about what you need to measure. So I've talked a bit today about analytes like total gene expression and cell surface protein expression, but I didn't touch on some of the other analytes that we can measure, like chromatin accessibility, immune repertoire profiling. So there's a lot of things that you can measure. And so thinking about which things you need can really help guide you as to which resources to start with. And this slide, it looks really busy, but the only point I want to make is that there are lots of different sample types that are being used today for clinical studies. And so you have a lot of options for how you can collect and handle and transport samples for single cell analysis. So for example, if you're working with blood, these can be collected in standard types of tubes, and they can be shipped at room temperature for short time periods or in the fridge. And while you do not want to freeze whole blood, you can flash freeze whole tissue samples and ship them on dry ice. So this is really just to say that there's a lot of different options based on what type of sample you are working with. And also, we have a huge number of resources available on our website and our support site to help you get started with your sample preparation. We have a lot of different webinars, both by 10 experts and our customer speakers who work with a lot of different sample types. And we have a lot of getting started tutorials on the support site and demonstrated protocols and tech notes. So if you have any specific questions about your samples, please don't hesitate to reach out to our really excellent support team. And I also just want to put in a plug for a new product that we're very excited about that we'll be launching around the middle of this year. And so I've told you about, all the different options for handling and transporting samples, but it's also true that one of the, big logistical challenges for doing single cell work can be the need for viable cells. And so you're sort of racing against the clock to get those cells into a stage where you can process them. And so we will be solving many of these challenges with our fixed RNA profiling kit, which will enable fixation in paraformaldehyde at the point of sample collection, which will really just lock in that fragile biology and remove those time constraints for working with these precious samples. So you don't need to rush to get them shipped to your service provider. And similarly, being able to fix these samples will help CROs be able to mirror a lot of the same processes they currently use for other techniques. So this will really, I think, streamline the ability to incorporate single cell studies into larger projects. So stay tuned to hear more from 10x in the next few months about this. And then last week, we launched a 10x Genomics certified CRO program, which is part of our global certified service provider program. Azenta was chosen by 10x to be one of the 3 inaugural global CROs. This program was designed specifically to support our pharma and biotech customers with qualified CROs for them to be able to execute these large-scale translational and clinical research projects. And we are so happy to collaborate with Azenta to be able to provide our mutual customers with additional support for these translational and clinical research projects, just for continued success. And so with that, I will hand it over to Azenta.
Thank you for joining our webinar today. I'm Bhagyashree Birla, a technical application scientist in the next generation sequencing team at Azenta Life Sciences. A little bit of background about who we are and what we do. Azenta Life Sciences is an organization that encompasses several different companies under one umbrella, including GeneWiz, which is what we were known as until December last year. Azenta Life Sciences provides sample solutions to accelerate research and discovery in both preclinical and clinical stages, as well as aiding with manufacturing and distribution of therapeutics. From what was previously our GeneWiz arm, we provide several different services, including plasmid preps, next-generation sequencing, Sanger sequencing, and PCR assays, as well as gene synthesis. Azenta is also a 10x Genomics certified service provider, trained on 10x Genomics best practices, helping you overcome roadblocks and giving you the confidence you need in your research results. In today's webinar, I'm going to discuss our single-cell sequencing services performed using the 10x Genomics platform, including some topics such as limitations to traditional bulk RNA sequencing assays, common sample types for single-cell sequencing and challenges associated with those, some case studies, as well as using 10x Genomics platform for immune repertoire and ATAC-seq and its applications. To highlight some benefits of single-cell RNA-seq versus bulk RNA sequencing assays, I'm going to use this analogy of a fruit salad and a fruit smoothie. Consider a complex tissue being similar to a fruit salad where we have several different fruits such as watermelon, strawberry, kiwi, apples, etc. that are adjacent to each other. With bulk RNA sequencing, we would dissociate this fruit salad and turn it into a fruit smoothie and try to identify which fruits are present in more or less quantity, such that after analysis we would be able to distinguish whether a smoothie has a lot more strawberry in it or if it has a lot more kiwi. But we would not be able to understand if there are different kinds of strawberries, kiwis or apples present in the same smoothie, or if there's a certain fruit such as a watermelon which is present in very low quantity. That would be difficult to detect. In contrast, for single-cell RNA-seq, we would sort these different fruits from the fruit salad into different buckets and analyze them separately to understand how many strawberries or kiwis or apples are present in the same sample. Applying this analogy back to a tissue sample using single-cell RNA-seq, we would be able to use this technique for profiling of heterogeneous cell populations as well as profiling of rarer cell types, example stem cells that may be present in the cell populations. With most bulk RNA-seq studies, a certain amount of RNA input is required for library preparation. On the other hand, single-cell RNA-seq works with a small number of cells, and thus it can be used for profiling of primary cells that cannot be cultured further. Here's an overview of the standard single-cell RNA-seq profiling workflow using the 10x Genomics platform at Azenta. Usually, the customer isolates cells using cell sorting or from tissues and freezes the cells using an optimized protocol provided to you by Azenta and then ships the samples to us. Once we receive the cell samples, we assess the cell viability as well as count the cell number to ensure that we have a sufficient amount to proceed with library preparation. In case the viability does not meet our QC requirements, we perform a dead cell removal protocol and then proceed with loading the cells on the 10x platform wells. On the 10x platform, each cell is barcoded, followed by the reverse transcription reaction and cDNA generation, preparation of libraries, and sequencing. Sequencing is performed on the Illumina platform, followed by analysis using the 10x Cell Ranger and Lupe browser. There are a few different sample types that can be used for single-cell RNA-seq. However, there are some challenges associated with the preparation of each of these samples so that they can be processed using the 10x Genomics platform. Some of the different sample types include cell cultures, fresh or frozen tissue, and fixed cells. For cell cultures, it is necessary that the cells have a high viability as well as a sufficient number of cells. We help customers accomplish this by providing a protocol for cryopreservation and checking the viability of cells upon arrival at our lab. In case the viability is lower than required, we perform a dead cell removal protocol using magnetic beads to increase the viability of the sample. With fresh tissues, our lab personnel have a lot of experience to perform tissue dissociation, which is required to isolate the cells. Similarly, with frozen tissue, which is ideal for already frozen samples, difficult to dissociate tissues, or fragile cell types, we perform a nuclei isolation process before proceeding with the single nuclei workflow using 10x. I'll speak a little bit more about nuclei isolation process in the next few slides. Methanol fixation is ideal for cells that are too fragile or sensitive for robust cell viability after cryopreservation. Customers can store their fixed cells at -20 or -80 degrees for up to 6 days. This eliminates post-thaw viability issues and transcriptional change concerns, though after methanol fixation, storage and rehydration, it is possible that only up to 50% of the cells are recovered. In case customers want to perform these procedures at their lab before shipping the samples to us, we also provide all the optimized protocols to you so that you are able to extract the cells for submission to us. However, one of the common challenges that we see with researchers is the unfamiliarity with the different sample types and having the resources to perform these different procedures. This is where our experienced lab professionals can help you to generate the ideal sample types for downstream single-cell processing. In addition, we also offer data analysis workflows, which can be a bottleneck for several researchers. These data analysis workflows include cell clustering and gene expression analysis using the 10x Cell Ranger and Lupe browser. For those worried about cryopreservation of their cells, we present an internal case study where one of our customers wanted to better understand response of cancer cells to a drug. Bulk RNA sequencing had previously been performed, but the investigator was interested in a potential heterogeneous responses to the drug. Briefly, a cancer cell line was exposed to either a drug or vehicle alone, and cultures were collected at time point 0, 24 and 48 hours. The nature of this time course experiment presented a potential obstacle since the investigator was concerned about batch effects due to processing the cells on different days. For this reason, the cells were cryopreserved after initial collection and stored until all the samples were available. Importantly, it has been shown very robustly that cryopreservation results have no statistically significant changes in the transcriptional profile. Here we highlight a few examples from the paper demonstrating that fresh and frozen cells have similar profiles. This research group tested many different cell types and tissues with consistent findings, suggesting that this protocol is applicable for a wide range of cell types. Here we present a case study where the research group were interested in characterizing the transcriptional differences between these three cell populations. The red T0 cells and green vehicle-treated cells cluster together at the bottom left. Cells exposed to the drug for 24 hours can clearly be distinguished, and cells exposed for 48 hours in orange can be distinguished even more. Thus, we were able to identify several different genes that were differentially expressed with statistical significance. We next turned our attention to the cells treated with the drug. These results confirmed what was observed by separate bulk RNA sequencing experiments. However, upon closer examination, a minor population of non-responsive cells were identified. These cells cluster in proximity to each other regardless of their treatment status, suggesting that these cells are refractory to the drug. Studying the differences between these cells and the responders will give us a better clarity to determine why some patients would not respond to the drug. As I mentioned earlier, there are a couple of challenges associated with processing fresh cells or tissue samples, such as lack of equipment, personal skill set, or time to perform tissue dissociation and cryopreservation. It is also a challenge to preserve cellular viability, as Traditional solutions such as tissue cryopreservation or methanol fixation may not be feasible. These solutions require specialized equipment such as tissue dissociators or a challenging workflow which must be performed quickly. A potential solution to some of these different challenges is the single nuclear RNA sequencing workflow. At Azenta, we have developed a workflow for processing frozen tissues where customers can collect the tissues at their clinical sites and ship them to us. Once we receive the frozen tissues, we prepare a nuclear suspension using enzymatic and mechanical dissociation methods and filter and pellet the nuclei with an optional sucrose gradient centrifugation step which is performed for certain types of samples, followed by QC of the nuclear suspension. Once the nuclear suspension passes our QC metrics, we would perform single nuclear RNA-Seq workflow through the 10x platform, which is quite similar to the single-cell RNA-Seq library preparation method. Based on this technique, we have been able to successfully demonstrate that nuclear isolation is possible from a variety of tissues, including but not limited to brain, liver, kidney, spleen, and colon. Our internal data suggests that we can detect greater than 2,500 genes with 50,000 reads per nuclei, as well as cluster the different types of nuclei to identify heterogeneous cell populations. At Azenta, we also offer a cell assay developed for profiling immune cells and VDJ analysis using the 10x Genomics platform. With bulk immune profiling, we are able to identify VDJ regions of B cells and T cells individually, but cannot tell if these regions are part of the same cell. This can be overcome using single-cell immunoprofiling, as using this technique, we are able to sequence all the way across the heavy and light chain and get it all together in one go. Thus, we are able to identify exactly which immune repertoire comes from which individual cell. 10x also provides the ability for us to use the Cell Ranger and Lupe browser for analysis post-sequencing. Using the 10x Genomics platform, we are also able to perform single-cell ATAC-seq. Applications for this technique include identification of chromatin accessibility regions to reveal subpopulation-specific regulation and detect altered gene regulation or novel enhancers in rare cells and subpopulations. We are also able to identify and classify diverse cell types present in heterogeneous cell populations and discover states for lineage and development tracing to understand mechanisms of cell fate. A comparatively newer workflow that has been developed by 10x is single-cell multiomics. Here, we split the same cell sample for simultaneous library preparation for ATAC-seq and RNA-seq, followed by sequencing and integrated data analysis to look at the results from both the assays together. To recap, Azenta provides end-to-end solutions for 10x genomics processing. This includes cell viability check, dead cell removal if necessary, cell partitioning and barcoding of the beads, library preparation with unique barcodes added for each individual cell. Sequencing is performed on the Illumina platform, followed by bioinformatics data analysis using the different 10x browsers to visualize results and also receive custom analysis. We know that you have a lot of options out there, but here are some reasons you should use Azenta for your NGS service needs. We do have a lot of experience working with a variety of next-generation sequencing techniques, not just limited to single-cell sequencing, as well as processing of many different sample types. We are proud to have one of the highest on-time project delivery rates in the industry, with over 90% of our projects being delivered within the promised turnaround time. We also have expertise working with a broad range of projects, including several different sample types and many different species. Azenta is known for superior customer service, including PhD level study managers that are available to support you at every step of the process, beginning with technical consultations for all of your project needs, as well as an advanced online platform for ordering and project management, and an extensive network of drop boxes so that you are able to submit samples at a nearby drop box at a convenient location for you. Under Azenta Life Sciences, we also offer a range of sample storage and management solutions in addition to genomics and data analysis. With our expertise as a certified service provider with 10x Genomics, you can access the expertise of a team of highly trained scientists and bioinformaticians to support your research. That's all for my talk today. Thank you, and if you have any questions or need further information regarding our next generation sequencing services, please feel free to email us at ngs@azenta.com. Thank you.
Great. Now I would like to thank both of our speakers for presenting great talks today. We're now ready to get started with our live Q&A session. Please remember to submit your questions using the Q&A box on your screen. We'll do our best to get to your question, and we'll get started right away.
Okay, so I have a lot of questions here. Let's go to Bhagya first. Can you please share the freeze and thaw protocol for the cryopreserved sample and which dead cell removal chemistry?
Yes, absolutely. We'd be happy to share the cryopreservation protocol with you, as well as the thawing protocol. Please email us at ngs@azenta.com, as we do have different recommendations based on your sample type. So we would discuss your project and then share the appropriate protocols with you. Regarding the dead cell removal chemistry, we use a magnetic cell sorting method to remove the dead cells from the samples before proceeding with the library preparation.
All right, thank you very much for that answer. For the next one, how many cells need to be collected for single-cell RNA-Seq?
So generally, we recommend collecting about 1 million cells to ensure that we have enough material, and also gives us an option to dead cell removal in case of low viability of the cell sample. However, this is an ideal requirement, and we have worked with much lower amounts successfully. So we can work with quite low number of cells depending on the sample type.
Thanks for that. All right, let's stay with Bhagya for a couple more questions before we go to Dina. What's the difference in using cells versus nuclei?
So there is a few different factors to consider when you're working with cells versus nuclei. If you're working with frozen tissue samples, we do recommend nuclei isolation as the preferred method. However, since we are assessing the transcripts in the nuclei only, the total number of genes that are detected may be overall lower than what we would detect from single cells. So it is necessary that whenever possible, it would be best to plan based on the project goal prior to sample collection, whether you need to collect a frozen tissue sample or whether single cells would be the ideal sample type for you.
And that kind of segues us into our next question for Bhagya. Are there specific requirements for amount of frozen tissues required for nuclei isolation?
So we generally ask for researchers to submit about 100 milligrams of frozen tissue. But again, this is an ideal requirement and not an absolute requirement. and we do have experience working with lesser amount of tissue as well. So if you do have a lower amount of tissue, we would encourage you to talk to our excellent technical team and assess the best single cell or single nuclear workflow for processing for you.
All right, thank you. So let's go over to Dina, and we'll go back to Bhagya in a few moments. So for Dina, are results from single nuclear RNA-seq comparable to those from single cell RNA-seq?
Oh, yeah, that's a good question. So, it depends on what you need to measure for one thing. And also, sometimes, if you're working with frozen tissue, for example, you have to work with nuclei. And so there will be differences in terms of what transcripts are in the nucleus versus the cytoplasm. And, but, we have plenty of information on our website about comparing the data and what you can expect. So I think both are very valid to work with.
All right, let's stay with Dina for the next question. For the new fixed RNA profiling assay you talked about, does the fixation step inactivate infectious samples?
Oh, yeah, that's a common question these days with a lot of people working on COVID-infected samples. Yes, so the fixative there is 4% paraformaldehyde, which is, widely accepted to, inactivate infectious samples, virus-containing samples, so that you can remove them out of a BSL-3 lab. So that is another advantage of working in that sort of environment. Yes.
Great. And let's go with one more question for Dina. What do you recommend in terms of FACS enrichment before single-cell gene expression experiments?
Yeah, so many of our users do some sort of enrichment, upfront to sort cells or to clean up a sample. So you can use FACS to remove contaminants like debris, or of course, if you want to enrich for particular populations of cells. And so, again, there's a lot of protocols we have online for working either with cells or nuclei after you do an enrichment. There's also magnetic bead enrichment protocols. So yeah, whatever kind of fits in best with your workflow, I would say is good to put in there.
Thanks for the answer, Dina. All right, let's go back to Bhagya. Do you have ability to use 10x Genomics' CellPlex multiplexing solution?
Yes, we do have experience working with 10x CellPlex solution. It would definitely depend on your project goals to determine if that's a good workflow for you. So definitely we would recommend technical consultation with our experts to figure out your sample type and project goals. And then we can definitely talk a little bit more about the CellPlex solution.
Great. Does Azenta also support site-seq cell hashing for 10x workflow?
Again, yes, definitely we do offer site-seq as well as cell hashing for the 10x workflow. It would depend, it is a custom solution at our end, and we'd be happy to chat with you and talk more about your project goals. But as an answer to your question, yes, we do offer it.
Thank you. Let's see here. So this person is interested in the software used for analysis. Can you expand on that?
Yes, absolutely. We use the 10x Cell Ranger and Lupe browser for the downstream single cell analysis. And some of the output that we get from this platform is the read alignment, feature barcode metrics, digital gene expression matrix, as well as gene expression analysis, etc. So all of these plots and charts that are generated from the browser are shared with you post-analysis.
Great, thank you. Let's see here. That's another question for Bhagya. For single-cell RNA-seq, do you run triple repeats for samples?
This is a good question. So we would run as many samples as you need. It's basically dependent on your particular experiment and the number of biological replicates that you would need for the particular project goals. We do not specifically run triple repeats for the samples, but we could do that based on what is needed for your experiment.
Right, thank you very much. Let's see here. let's stay with Bhagya for a couple more questions here. If we are interested in running treated cells for analysis, what considerations would there be for sample preparation?
That's a good question. So there's a few different considerations for running treated cells. First of all, this method is ideal for cells that are too fragile or sensitive for robust cell viability after cryopreservation. So a certain type of cells we do recommend to go forward with this protocol. It is also important to note that we need a very high number of cells to start with, because after methanol fixation, the cell recovery rate post rehydration may be about 50%. And also, so far, this protocol has been validated for single cell 3' gene expression workflow only. So it may not be compatible with 5' gene expression or single cell immunoseq. So some of these different factors should be considered before you think about proceeding with the methanol fixation protocol for your sample types.
Great, thanks very much. Let's see here. This is a question regarding sample type compatibility, looks like. Can your protocol be used for plant protoplast cells?
If this is a question for cryopreservation protocol, that is something that we would need to check because our protocol works with most eukaryotic samples. With protoplast cells, I believe it should still work as long as the cells do not have a cell wall. But we would need to look further into the species type that you're working with and the exact cell type before we are able to make that recommendation. So definitely, I would encourage you to connect with our team and we'd discuss your project in detail before we recommend the appropriate protocol with you.
Thanks very much. There's a couple more questions here for you, Bhagya. If we have an option of choosing between cryopreserved cells and methanol fixed cells as sample type for single cell RNA sequencing, which method would you recommend?
Right. So again, the method would be dependent on a few different factors, such as cell viability post-thaw and the total cell number available. So in most cases, I would recommend cryopreservation of live cells. However, as I mentioned, there may be some cell types that may be too fragile or sensitive to the cryopreservation protocol, and that's when we may need to go through the methanol fixation protocol for single cell RNA-seq. It is important to remember that a large number of cells would be needed for the methanol fixation protocol.
Excellent. Thank you, Bhagya. Are there any limitations on the species type for single cell sequencing?
So as such, there's no limitations in the eukaryotic species that we can work with for single cell sequencing. The most common species that we have worked with for single cell processing is generally human or mouse. But that's just because there's several different applications of this technology in areas such as immunology or cancer research and drug discovery. But to answer the specific question, these are eukaryotic cells as long as they have the general profile of eukaryotic cells with a cell membrane. We should be able to proceed with single cell sequencing for it.
Thank you. Let's see here. Let's go to Dina, because that kind of segues into another question here. For Dina, is it possible to measure neutrophils from blood? I've heard it's not possible to do single cell analysis on this type of blood type or this cell type.
Yes, it is possible. So the important thing to keep in mind if you want to analyze neutrophils is you have to keep your sample at room temperature until you're ready to process it. And ideally, you want to process it fairly quickly. So, you might have heard that neutrophils don't like cold temperatures, and they tend to lyse if you put the sample on ice or freeze it. So we do have a demonstrated protocol on our website specifically for neutrophil isolation. And we also have some other information for how to analyze data with neutrophils, because we know there's a lot of people who are interested in studying
studying those in blood or in other tissues as well.
Awesome. Thanks very much, Dina, and thank you, Bhagya. So we are at the top of the hour, and that wraps up our webinar for today. Thank you again to our speakers for joining us for the great talks and the Q&A session. And thank you to our attendees for some great questions today. A recording of this session will be made available on demand shortly, and you'll receive an email when that is available. If we didn't get to your questions today, or if you have any follow-up questions for us, you can contact support at the email address support@10xgenomics.com. And in a few moments, you'll see a survey pop up on your screen. We really appreciate any feedback you can provide so we can learn more about your research and how we can improve these webinars. Thank you all again, and have a great day.